Hello everyone, a warm welcome to our channel, that is the Progressive M.L.T. If you are completely new, just subscribe our channel. Today's topic going to be very much interesting, that is the Corynebacterium diphtheria. We are going to understand about the Corynebacterium diphtheria. We are understanding the diphtheria. You will lots of time you have already listened, that is the diphtheria. Now we are understanding the particular things. Let's start the lecture by understanding Corynebacterium diphtheria. Okay, let's see. First one is the basic introduction we need to understand about the Corynebacterium diphtheria. That is it is a gram-positive bacteria and non-motile. Means they are not motile. Okay, and see here you will find out that they are non-spore formation. Means they are not doing any kinds of spore formation. Okay, and they are rod-shaped bacterium, or you may say that that is a bacilli. Okay, and it causes the diphtheria. What is the diphtheria? Diphtheria basically one type of upper respiratory tract infection. There is a two type of respiratory tract. One is the upper respiratory tract and another one is the lower respiratory tract. This diphtheria basically causes the upper respiratory tract infection. Okay, who discovered the Corynebacterium diphtheria? See here, first one is the Klebs and Loefflers. Okay, and that is a part of the normal flora. That is a part of the normal flora in skin, but who are causing that toxin as well as who are releasing that toxin, they basically causes the Corynebacterium. Okay, uh, now we are understanding the morphology of the Corynebacterium diphtheria, how it look like. See, we have already understood that the Corynebacterium is Chinese letter shape. You might lots of time listen into the, whenever you are understand about the normal morphology of the bacteria, you might lots of time listen that that is the Chinese letter shape. See here, that is a Chinese letter shape. You will find out Chinese letter, how it look like. It might be V. See here, that is a V, or it might be L. Okay, V or L it might be. So that it's a Chinese letter, and that is the gram positive. I have already told you that. And spore, that is the spore is negative. They are not spore forming. Motility, non-motile. They are not doing any kinds of motility. And the capsule, that is a not capsule present. There is a, they are non-capsulated bacteria. Okay, and we are doing the Albert staining. Albert staining is, what is the Albert staining? Green bacilli with bluish black metachromatic granules, we will find out in the Albert staining. That is a special staining we are doing for the Corynebacterium diphtheria. This stain, you will find out lots of questions are coming from the particular stain, that this stain we are doing for understanding which bacteria, then you have to choose the Corynebacterium diphtheria. Okay, after that, now we are coming to the culture characteristics, means how to we grow the Corynebacterium. We need to understand, right? See, that is the aerobic and facultative anaerobic. Aerobic means with oxygen they can also grow. Facultative anaerobic, they can also grow without oxygen as well. Okay, temperature, that is 37 degrees centigrade temperature, and optimum pH, that is a 7.2 to 7.6. Slightly alkaline pH is required. Okay, and the common media, you might take the Loeffler serum slope, that is one type of enriched media. You may also take the blood agar, that is also enriched media. You may take the tellurate blood agar, you will find out the black or gray colony due to the tellurate reduction. That is the selective media for the Corynebacterium diphtheria, that is a blood tellurate agar or tellurate blood agar. Okay, that's all about the culture characteristics. Now we are understanding about the biochemical test, because we have already understood the morphology, already understood the culture characteristics. Now we are going to understand about the biochemical test. If you just want to take the PDF, or if you want to just join in the medical lab technology, then just check our description box. You will find out everything. There is a test, there is mock test, there is the PDF of notes, there is the lots of valuable content. You will find out completely free of cost. There is the WhatsApp group you may join, you will find out this PDF as well. Okay, as well as the vacancy update, you will find out from the particular group. All kinds of M.L.T solution, you just check our description box, you will find out it. Okay, see, first one we may do the catalyst test, that is positive, oxidase test, that is also negative, urease test, that is variable, nitrate reduction test, that is positive, and the glucose and maltose test, that is positive, sucrose test, that is variable, and motility non-motile. If you are completely new into our channel, just check the biochemical test classes. You will find out that how do we do these kinds of biochemical tests, in which test cases they are positive. Okay, I have already taken the class of biochemical tests. Look down upon the class. Okay, now we are coming to understand that is a pathogenesis, means how the disease is spreaded, how the disease is appearing. That is portal of entry. You need to understand that is a coughing, sneezing, that's all. See here, that is a coughing, sneezing, that's all are the portal of transmission. As well as direct contact, they may transmit it. Direct contact means from the lesion, from the contaminated object, they basically transmit it. Portal of entry should be nose, throat, skin, wound, conjunctiva. They may easily spread it by this kind of portal. Okay. Now we are understanding that there have some kind of Corynebacterium diphtheriae. They are not producing the toxin. Who are producing the toxin? They causes that disease. Only toxigenic strain causes disease. Who are producing the toxin? They only causes the disease. Okay, diphtheria toxin produced by the toxin. Getting my point? Carried the bacteriophage. Bacteriophage basically carries the toxin, which releases the toxin, and which releases the diphtheria toxin. They basically causes the diphtheria. Okay, they basically inhibit the protein synthesis and cell wall become death. Whenever the protein synthesis become decreases, cell wall become death, then the disease might be occurred. Okay, that's all are the thing regarding the pathogenesis. Okay, now we are understanding which kinds of symptom might be occurred. Firstly, sore throat, because that is the upper respiratory disorder, so that sore throat is the common. Fever, malaria, and you will find out that thick gray pseudo membrane as well as into the pharynx, a ball neck appearance into the lymphadenopathy, you will find out. Okay, airway obstruction, you will find out. Cutaneous diphtheria, you will find out the chronic ulcer. Okay, and systemic toxic effect, you will find out the myocarditis, you will find out the neurotitis, you will find out the kidney damage. Into this condition, diphtheria might be occurred. Okay, that's all are the clinical significance. Now we are coming to the lab diagnosis. We have already understood the lab diagnosis, as we have the knowledge that lab diagnosis means we have to collect the sample. From where we are collecting the sample? Where the infection is occurred, as well as from where we can easily identify the disease. We have to collect the sample from there. Okay, see, first one is the sample collection, that is a throat swab we may collect, nasal swab or listen swab we may collect. Okay, microscopy, please understand the gram staining, that is a gram positive cup shaped rod we will find out. Albert staining we may do green bacilli with the bluish black granules we will find out. Okay, we have already understood it. And loeffler serum slope, tellurate blood agar, we have already understood that this kind of culture media is used for the Corynebacterium diphtheria. Identification biochemical test, we have already understood that this is the special test, Elix gel precipitation test for the toxigenicity test, or diphtheria toxin is present or not. Okay, as well as we may do the PCR as well as the ELISA for identifying the toxin gene detection. Okay, that's all are the thing regarding the Corynebacterium diphtheria. Now we are already coming the last thing, that is the diphtheria vaccination you may do for preventing the diphtheria, and that is the last thing. Thank you so much for understanding, thank you so much for listening. Just subscribe our channel and share with your friends, colleagues, seniors, juniors, and if you want to join with us, there have lots of benefits you will find out. That is your ultimate M.L.T solution, PMLT. Just check our description box, you will find out everything. Thanks a lot for listening.